Okay, so now we are going to look at the types of RNA interference. So number one, we have short interfering RNA and then microRNA. So these are the two types of RNA interference. So now we are starting with short interfering RNA. So the short interfering RNA is the commonly used RNA interference tool for inducing short-term silencing of protein coding genes. So the short interfering RNA is used in silencing gene for a short term. And we have different types of gene regulation. We have short term and long term. Short term is like when you silence a gene, or when you stop, or when you block the expression of a gene for a short period of time. While for a long-term gene silencing is a type of regulation in which the expression of the proteins or the expression of the genes is blocked for a long period of time. So the short interfering RNA is the RNA interfering tool for short-term gene silencing. And then this short interfering RNA is double-stranded RNA molecule. It means that it's made up of two strands. These two strands are made up of sense and the antisense strand. The antisense strand is called a guide, while the sense strand is called a passenger. So these double-stranded short interferon RNA molecules, it interferes with a gene expression by degrading the mRNA. So it means that when once the mRNA is being degraded and destroyed, then the protein translation will stop. So that is the role of short interferon RNA. It causes a short-term gene silencing. It is double-stranded, and then it targets the messenger RNA by degrading it. It will digest it, cut it into smaller molecules, and destroy it. And then once the mRNA is destroyed, it means that there is no protein synthesis. The protein synthesis will not take place. Okay, then the structure. The structure of the short interferon RNA, as I said, is double-stranded. It's made up of sense and the antisense strand, which make it to form a duplex, and then it's made up of 20 to 24 base pair lengths. So it means that this short RNA interference is made up of 20 to 24 nucleotides. So these are the nucleotide sequence that are found in the short interferon RNA. So it means that it is a short, and then it's made up of an overhang. It's made up of an overhang. These are the two overhangs. We have it at the 3' end at this direction, and also at the 3' end at the sense strand. So this is the structure of short interferon RNA. It is double-stranded. It's made up of 20 to 24 base pair, and is also made up of overhang. Now then, the next thing that we are going to look at is the mechanism of the of the short interferon RNA. So what usually happens, the short RNA interference is double-stranded. It is double-stranded. So then the first thing actually is an enzyme called a dicer, and these dicer enzymes cut or clip this messenger RNA into smaller fragment. And then an argonate, which is an enzyme, will now unwind this short double-stranded DNA. It will now open it off to form two strands, which is the antisense strand and the sense strand. And then this argonate, once it unwinds the short interfering RNA, then it now forms a complex with the antisense strand. And the complex is called RNA-induced silencing complex. And this RNA-induced silencing complex find their complementary mRNA. So the RNA-induced silencing complex is always complementary to the target mRNA. And then once this RNA-induced silencing complex recognize or identify the complementary RNA, and then it will now bind to it and induce the cleavage. Then it will now cleave it. It will now induce its cleavage. So therefore the mRNA will be cleaved. Then the mRNA is now caught and recognized as an abnormal by a cell. And this causes a complete degradation of the mRNA. And in turn, the translation of mRNA into amino acid and proteins will be blocked, thus result in silencing the gene that encode the mRNA. Okay, then now let's look at this. This is a diagrammatical representation of the mechanism of the short interfering RNA. So generally, as I said, the double-stranded RNA, this double-stranded RNA of short interfering RNA is made up of 20 to 24 base pair, and it can be in the form of hyphen. And then an enzyme called dicer will now act on it and cleave on it to form short double-stranded short interfering RNA. And then an argonate, which is an enzyme, will now often unwind the double-stranded short interfering RNA into sense and the antisense strand. And then the sense strand will now form a complex with argonate to form what we call RNA-induced silencing complex. And then this RNA-induced silencing complex will now recognize the complementary mRNA and bind to it, and then cleave it. And once it cleaves it, then it now silences the genes. So this is the mechanisms of short interfering RNA mechanism. Sorry, of RNA interference. So therefore, in a simple term, from this diagram, we can say that the short interfering RNA is a type of RNA that is usually long, cut into a short double-stranded RNA with an enzyme called dicer, and then an argonate will now unwind the DNA to form sense strand and the antisense strand. The argonate will now bind with the antisense strand to form a complex called RNA-induced silencing complex, which is complementary to the target mRNA. Then it will now bind to the target mRNA, cut it. Once it is cut, then it will now recognize as an abnormal RNA. Then it will now be digested and degraded. Therefore, the protein synthesis will be blocked. So that is for the short interfering RNA. So therefore, now we are going to stop for this, and then we'll continue with the microRNA.